So good morning, everyone. Today, the topic of my presentation is shotgun sequencing. So let's start with the topic. Now, while we are starting with the topic, let's give a brief introduction about shotgun sequencing. So this has been developed for sequencing large fragments of DNA in 1979, and it was the initiative for full genome sequencing. The chain termination method of DNA sequencing, which is also known as the Sanger sequencing, can be used only for short DNA strands of 100 to 1000 base pairs. Due to the size limit, longer sequences are subdivided into smaller fragments that can be sequenced separately, and then these sequences are assembled to give the overall sequence. So this is something what we call as shotgun sequencing. Therefore, shotgun sequencing is an approach that is used to decode a genome by shredding or shotgunning it into smaller fragments of DNA, which can then be individually sequenced. Then the sequences of the fragments are ordered based on overlaps in the genetic code, and finally it is reassembled into the complete sequence. Now over to some advantages and applications of shotgun sequencing. First, dealing with the advantages. So by removing the mapping stages, whole genome shotgun sequencing is a much faster process than clone by clone sequencing, as because in clone by clone sequencing, what was done is uh, an entire map of a particular chromosome of the genome was made first, and then the DNA was fragmented into smaller fragments, and then it was sequenced. So, making a map first and again breaking it down and again sequencing, the entire process would have taken a much longer time. So if we remove the mapping stage and directly go for fragmenting and then sequencing them and then assembling them, that will be much easier. So whole genome shotgun sequencing is something that is much faster than clone by clone sequencing. Also, whole genome shotgun sequencing, it uses a fraction of the DNA that clone by clone sequencing will need. It is particularly efficient if there is an existing reference genome. It is much easier to assemble the genome sequence by aligning it to an existing reference genome. Shotgun sequencing is much faster and it's less expensive than methods requiring a genetic map. Now over to some applications. It is used to remove errors, fill in gaps, or correct parts of the sequence that were originally assembled incorrectly when clone by clone sequencing was used. As a consequence, the reference human genome is constantly being improved to ensure that the genome sequence is of the highest possible standard. So this is a diagrammatic representation of shotgun sequencing, like have been mentioned previously. Uh, this shows that a large DNA molecule, this is fragmented to give smaller fragments of DNA, and then the fragments are individually sequenced, and then the sequenced are matched based upon the overlapping parts of each of the sequence. Like in the figure, three such sequences have been highlighted with purple, blue, pink, purple, and blue colors. And here we can see that the ends of these three sequences have some of the other portions that are overlapping or common. Like with the pink and the blue portion, we have GTTA in common, and with the blue and the purple one, we have CATA in common. So matching with these overlapping sequence, the entire sequence is assembled, and the last line shows here the assembled sequence, which have all the three fragments that are aligned together. This was just a flowchart or a representation of how this is basically done, shotgun sequencing. Now, over to some of the types of shotgun sequencing techniques that we are using, namely paired end sequencing, hierarchical shotgun sequencing, metagenomic shotgun sequencing. Now, coming to pair-end sequencing. So, how do we approach this pair-end sequencing? To apply this, a high molecular weight DNA strand is sheared into random fragments, usually 2, 10, 50, and 150 kb, and then it is cloned into an appropriate vector. Now we get clones. Clones are then sequenced from both the ends using chain termination method, yielding two short sequences. And each sequence that we get is called an end read or read 1 and read 2. And if we are getting two reads from the same clone, then it is referred to as mate pairs. Like we see in this diagram below, we have an insert, we have an adapter. This is an entire sequence and we have two reads here, read 1 and read 2, or these are called the end reads. Okay. Also, in the diagram which is mentioned in the top, we have paired end sequencing and we have mate pair sequencing. So this was the cloning and this entire process using vectors and then the chain termination method. And we can, other than that, we can also do this by fragmenting the genomic DNA. We have these fragments and then we ligate adapters to these, and these adapters are meant for sequencing. And then we pool it into a particular apparatus that is used for sequencing. We have cluster generation there, and further we go for sequencing. Next, for mate pair sequencing also, we fragment the genome, we get certain fragments, we bioannihilate the ends, and these bioannihilated ends, they get circularized, and the two ends, they come adjacent to each other. Next, this is fragmented, and we purify the bioannihilated fragments. So these are the enriched bioannihilated fragments, and we ligate the adapters. And then again, we go for cluster generation and we go for sequencing. So we have read the sequences. Now, how do we assemble them? The original sequence is reconstructed from reads using sequence assembly software. So first, the overlapping reads are collected into longer composite sequence, which are known as contigs. Now we can see from this diagram, which is shown on the right-hand side, these three dark arrows which are shown, these are three contigs. So contigs are nothing but contiguous sequences, which are stresses of the overlapping sequences. So contigs can be linked together into scaffolds. Now scaffolds are even the larger version of contigs, like they are showing the genome on a larger scale. Contigs are linked together to give scaffolds. So like it is shown in this figure below, three contigs or these three dark arrows together making a scaffold. So it's written below, a scaffold of three contigs, that is the thick arrows held together by mate pairs. And about mate pairs I have told in the previous slide. So contigs can be linked together into scaffold by following connections between mate pairs. The distance between contigs can be inferred from mate pair positions if the average fragment length of the library is known and it has a narrow window of deviation. Now, depending on the size of the gap between these contigs, different techniques can be used to find the sequence in the gaps. If the gap is small, that is 5 to 20 kb, then we can use PCR to amplify this region, followed by sequencing. If the gap is large, that is greater than 20 kb, then the large fragment is cloned in special vectors such as bacterial artificial chromosomes or BAC, followed by sequencing of the vector. Now, as we can see in this part, on the top part of this diagram, like it's briefly telling that we get reads, then we have overlapping sequences, then we go for local multiple alignment, alignments scoring, then we go for contigs. Contig give rise, like the contigs together, they give rise to scaffolding, and scaffolding will again give rise to finishing. That is, we come to know the sequences that we wanted to know of the genome. Next, coming to some pros and cons. Now, the proponents of this approach argue that it is possible to sequence the whole genome at once using large arrays of sequences, which makes the whole process much more efficient than more traditional approaches. But the detractors argue that although the technique quickly sequences larger regions of DNA, its ability to correct link between these regions is suspect, particularly for genomes with repeating sequences. As sequence assembly programs become more sophisticated and computing power becomes cheaper, it may be possible to overcome this limitation. Now we are coming to hierarchical shotgun sequencing. So the amplified genome is first sheared into larger pieces, 50 to 200 kb, and then it is cloned into bacterial host using BACs or PI-derived artificial chromosome PAC. Because multiple genome copies have been shared at random, the fragments contained in these clones will have different reads, and with enough coverage, finding a scaffold of back contigs that cover the entire genome is theoretically possible. This scaffold is called a Tilling path. Now, this one we come to know from this figure. We have two figures, A and B. Now, the figure shown by B is the one that is pointing out this hierarchical shotgun sequencing. So we have the genome that is first sheared into the larger fragments, and then it can be cloned into a host. And because multiple copies have been sheared at random, the fragments contained in these clones will have different ends. So these are the fragments which can have different ends and they can have enough coverage. So finding a scaffold of back contigs that cover the entire genome is theoretically possible. This scaffold is called a Tilling path. And the figure below here shows this Tilling path with a back library. So once this Tilling path is found, the bags that form this path, they are sheared at random into smaller fragments and they can be sequenced using shotgun sequencing on a smaller scale. Although the full sequencing of the bag contigs is not known, however, their orientations relative to one another is known. See, we have different lines in black and red, which are showing their orientations relative to each other. So there are several methods for reducing this order and selecting the backs or the back contigs that make up a tilling path. The general strategy involves identifying the position of the clones relative to each other and then selecting the fewest clones that are required to form a contiguous scaffold that covers my entire area of interest. Now this blue line is showing my area of interest. Now, the order of the clones is deduced by determining the way in which they overlap. Now, overlapping clones, how do we identify them? Which are the clones that are overlapping? Overlapping clones can be identified in several ways. A small radioactively or chemically labeled probe that is containing a STS or a sequence tagged site can be hybridized onto a microarray upon which the clones are printed. In this way, all the clones that contain a particular sequence in the genome are identified. The end of one of these clones can then be sequenced to yield a new probe, and the process repeated in a method which is called chromosome walking. But usually these walking they take a long time, so they are usually not opted for that much nowadays. Next, with the pros and cons of hierarchical shotgun sequencing. So because it involves first creating a low resolution map of the genome, hierarchical shotgun sequencing is slower than the whole genome shotgun sequencing, but it relies less heavily on computer algorithms than the whole genome shotgun sequencing. Again, the process of extensive back library creation and tilling path selection, however, makes hierarchical shotgun sequencing slow. A back contig that covers the entire genomic area of interest makes up the tilling path and it may be labor intensive. But now that the technology is available and the reliability of the data is demonstrated, the speed and cost efficiency of the whole genome shotgun sequencing has made it the primary method for genome sequencing. Next, coming to another form of shotgun sequencing, that is metagenomic shotgun sequencing. So, what is this all about? having reads of 400 to 500 base pairs length is sufficient to determine the species or the strain of the organism where the DNA comes from, provided its genome is already known. By using, for example, a KMR-based taxonomic classifier software, though I didn't mention details about the KMR, but this is a kind of a software that we can use for this classification, which I know theoretically. With millions of reads from next generation sequencing or NGS of an environmental sample, it is possible to get a complete overview of any complex microbiome with thousands of species like the gut flora. Advantages over 16S rRNA amplicon sequencing are, so how is this metagenome shotgun sequencing advantageous over 16S rRNA amplicon sequencing? Not being limited to bacteria, strain level classification, where amplicon sequencing only gets the genus, and the possibility to extract whole genes and specify their function as part of the metagenome. The sensitivity of metagenomic sequencing makes it an attractive choice for clinical use. It, however, emphasizes the problem of contamination of the sample or the sequencing pipeline. So this can be a problem. Other than that, it's quite advantageous over the 16S RNA amplicon sequencing, particularly in case of microbiology. Now, the newer sequencing technologies. The classical shotgun sequencing was based on Sanger sequencing method, and this was the most advanced technique for sequencing genomes from about 1995 to 2005. Now, the shotgun strategy is still applied today. However, using other sequencing technologies such as the short read sequencing and the long read sequencing. Okay. So, short read or next generation sequencing, it produces shorter reads anywhere from 25 to 500 BP base pairs, but many hundreds of thousands or millions of reads it can give in a relatively short time. Like if I order it in a day, on the order of a day, the output is quite high. Though the reads are shorter, but within a particular span of time, like within a day, I'm getting thousands and millions of copies. So this results in high coverage, but the assembly process is much more computationally intensive. So if we are aiming for these techniques, the assembly process is more reliable on extensive softwares and better computational techniques. So that has to be sophisticated in order to use these technologies are vastly superior to Sanger sequencing. Now, Sanger, which I've mentioned in my first slides. So these technologies are obviously much more superior to Sanger sequencing due to the high volume of data. High volume of data and the relatively short time that it is taking to sequence a whole genome. Now, these are the diagrammatic representations of short reads and long reads. So you have reference genome and we sequence, we break the genome into fragments, we sequence them, then we align or assemble them, and then we find certain gaps if we compare it to the reference genome. So with short reads, missing sequence data leads to gaps in the genome coverage, and it may limit variant detection. So somewhat better than this short read is a long read, where you see we have a reference genome. If we are comparing that to my target gene. So first of all, I fragment my genome into longer fragments. And so here the gaps are less, or I see no gaps. So long reads map uniquely, and it spans large variants, providing comprehensive variant detection. So somewhat better than the short reads. These are some of my references, and with this, I come to an end of my presentation with shotgun sequencing.
Thank you, Doutrina. Any questions?
Good morning, sir.
Morning.
Doutrina, could you please
No,
explain the
you're
term
not audible.
adapters? Hello, am I audible?
Yeah,
Yes, but
Monica,
it's too
audible.
low.
Yeah. Volume is low, but audible. Yeah.
Could you please explain the term adapters?
Adapters, wait, like these basically help you to, I'm telling you, wait. These are the sequence adapters. So after you make a library, these are my target fragments, DNA fragments. You ligate adapters to them. So now when you are inserting them into the flow cell, they basically adapters in one word, they can help you if you are going for cloning, they can help you to attach to the vectors. They can also act as binding site for primers if I have to sequence my target DNA. So these are the functions of adapters.
Can adapter act as an amplification for fragments?
Yeah. Yes, it can also, because see the primers, we need primers for amplification, amplifying a DNA. So DNA polymerase cannot itself start the amplification, it needs a primer. So adapters can act as binding sites for primers also.
Okay, thank you.
Yeah. These are basically oligo sequences. So these
Yes,
are basically
oligo.
oligonucleotide sequences. And most of these sequences are specific to the machine that you use. Like if you are using a Illumina, Illumina will have its own adapters.
Yes.
So they are machine specific, and they are some at the time, like for Illumina, you have multiple adapters available. Like you have AmpliSequence, Nextra, and many others. So basically, depending upon what you want to do, you have to choose an adapter, and the adapter uh, probably are mostly machine specific.
Okay, sir.
But these are sequences, oligonucleotide sequences.
Okay.
Sir.
Anything else? Do we have any other question from any other person? Because this is something which is new for almost all of you, I know. Though BHU has two next generation sequencers and IMS has a capillary Sanger sequencing machine. But I'm sure that most of you would not have seen or used that machine. So I expected a lot of questions coming out.
My next question is
Good morning, sir.
Hello.
Sir,
Yeah.
good morning, sir. Sir, a link has been provided on YouTube, or we can from where we can collect all the recorded PPT.
I'll send the YouTube link. I'll share the YouTube link on email. So from where you can get the previous recordings. And this presentation also will go on YouTube by evening today.
Okay, thank you, sir. Actually, I missed the initiation part of the presentation.
No problem. Dr. Tohina, I would like to have your comments.
Yes, sir. Good morning, sir.
Morning, Tohina. Good to see you.
Yes, I just somehow missed the previous two because of the timing. I intended to join, but I forgot. But this time I made it, and it was a nice presentation, Doutrina, and I think it was a very clear presentation. So yes, it is true that these are emerging concepts and we are not very clear with it because most part of it is outsourced, because even though we are having these facilities, but we are not very expert or competent with it presently. And once it is outsourced, we just get the analyzed data, and so we at many times do not know the details of it. So I will just ask, or I will just take a suggestion from Doutrina. If suppose in a resource limited setup, we want to study gut microbiome for some purpose, what will be your approach? Whether it will be a metagenomic shotgun sequencing or a 16S preferably.
Doutrina, did you get the question? Hello?
Just your view. I'm not asking a theoretical question. What should serve the purpose?
Where is Doutrina gone? It seems she have been logged out. Uh, Basically, I think she has been logged out because of some probably internet problem. Uh, if we see, the best would have been a shotgun sequencing. I see a lot of people are doing metagenomics nowadays. I think there are 3 or 4 theses which are registered with microbiology. And Doutrina herself is also doing what we call as estrabolome or estrogen dependent gut microbiome. So what she is using is because of limitations that we have in funding and infrastructure. What she is using is she's using a 16S barcoding, and she's going to use the capillary sequencer which is available. So this 16S barcoding basically what it does is it again does the shredding into smaller base pairs of 400 to 500, sequence them, and then use a software to identify the species, that which bacteria are we going to talk about. But then the problem of 16S is that it is limited only to bacterial species because we are using
Yes,
bacterial
exactly.
16S. If we go for shotgun, we can also identify the other than bacterias like fungus or the viruses, which usually you will not identify in 16S barcoding.
Yes.
So whole genome sequencing or a shotgun sequencing or any of the methods of a whole genome sequencing like next generation sequencing, especially long reads, because all these bacterial, fungal, and viral genomes are not very big genomes. So we can do long reads on it. They are not like human genome. The problem of human genome is that it is too large.
Hello.
Haan, Doutrina.
Yes, sir. There was some
You,
problem with the
yeah.
network, so I
Did you
was
hear? Did you hear Madam's question?
no.
Okay, I answered it. She basically wanted to know that if you have to choose a technique for metagenomics
Yes,
or
sir.
metabolomics,
Yeah.
what will you use? A 16S rRNA or sequencing? Whole
So
genome
like
sequencing.
sequencing can be obviously a better option
Yes.
because till 16S we are coming to know till the genus level, and for sequencing like metagenomic it can tell up to the species level. So that can be a better option to become more specific.
Yes, it is always a better option, but our problem is that we have limited resources. And though
Yes.
we have 2 next generation sequencers, but I have tried and talking to people, but uh, I'm not able to get them to do the sequencing for us till date. So if we outsource, then the per sample cost is so high
Yeah,
that
yes,
that it
that you
is
told us.
not possible for us to without the funding.
Yeah. So till 16 is also we can identify, but like that is till the genus level identification.
Yes, but then you will only identify the bacterias.
Bacteria, yes.
Yes.
So other gut microbiome you will not be able to identify.
No. Yeah, that is restricted.
So that is one of the shortcomings of using a 16S barcoding.
Yes, sir.
This is what you are planning to do in your thesis.
Yes, sir. Yes, yes.
It is going to be a shortcoming, but then this is the best that you can do within our institute without having to go anywhere else and trying to get the